The catalytic cycle of an enzyme action can be described in the following step. Arrange them in sequence accordingly. Catalytic cycle. Okay. Catalytic cycle. This catalytic cycle indicates that how the substrates binds to the enzyme and it catalyzes the production of product, is it? So in this process, from substrate to product, there are events, and those events which takes place, okay, this is known as catalytic cycle. So here the different steps, different event have been given, and this event have been divided into 4 different parts, and which one will come first, which one will come next, and which one, here options are given, all right? That first is here the events are A, B, C, D, but these events are haphazardly written, and what are those events, they should come in which sequence. Okay, this is the sequence that the first one, the binding of substrate induce the enzyme to alter its shape, fitting more tightly around the substrate. The substrate bind to the active site of the enzyme, fitting into the active site. Enzyme release the products of the reaction, and the free enzyme is ready to bind to another molecule of the substrate, run through the catalytic cycle once again. The active site of the enzyme, now in close proximity of the substrate, break the chemical bond of the substrate, and the new enzyme product complex is formed. So which one will come in the correct sequence? Okay, which one? Hello, hello, is there anyone?
Sir, is it number 2?
Is it number 2? Sure. Is there anyone else beside you? Hello, hello, Shavnath. Shavnath. Hello, Samrat. Okay, This is number two. Actually, when these enzymes, say if I draw that this one is the substrate and there is one portion that the enzyme, then this substrate come close to the enzyme, and enzyme is always have a uh, enzyme always have some special places, and this is very specific. This cavity is called the active site. Okay, this cavity is called active site. This cavity is made of amino acid which are very special, some special amino acids present in the enzymes. They make a cleft-like structure, and this cleft-like structure or pocket-like structure is the active site. The substrate come close to the enzyme and it fits to the active site. Okay, once it fits to the, say this will fit to the active site exactly in this way. All right, say now the substrate it has changed and it has fit to the active site. There will be close association, and initially it is loosely bound, after that it is tightly bound. This is the binding of substrate. First is substrate bind to the active site of the enzyme, fitting into the active site, and next that it induces enzyme to alter its shape. Okay, this enzyme slightly after binding, this enzyme slightly its shape, alter its shape, fitting more tightly around the substrate. Next, actually active site is not a rigid structure. This is little bit versatile. It can change its shape. Okay, it is flexible. So it can change its shape. So and then tightly fit to the around the substrate. And after that, whatever next step is that this bond, a specific bonds of the substrate, they may break, they may change, or new bond may be formed. Those all those things takes place. So the substrate change into a product, right? The substrate then change into a product. So now it is the enzyme substrate enzyme product complex, and after that the enzyme and the product is separated. This enzyme is free now to join again to a substrate. So that the substrate and enzyme combine, there is a lot of close association. After that, this forms the enzyme substrate complex. Then In the substrate, there is a change, and the substrate is changed into product. This change takes place by formation, maybe by formation of new bond, or maybe by the breakdown of the bond. Okay, so this enzyme product is produced, enzyme product then separate, and the enzyme is free to carry out a new enzymatic catalytic cycle. Okay, so here it is the B, after that A, then D, C. So this one is the answer. Next one, activation energy. I think we have learned the energy of activation or activation energy. Here, remember that is activation energy or energy of activation. Energy of activation. This energy of activation is the energy which is required for the progress of the or for initiation of the chemical reaction. To carry out a chemical reaction, that certain amount of energy is required. Okay, so this is here that the activation energy, what is the actual definition of the activation energy? This is the difference in the average energy content of S. S stands for substrate, from that of a transition state. Transition state is the short transient, okay, short-lived stage. This is substrate, and then enzyme is there, enzyme get, and there is a condition where this more there must be increase in the energy level of the substrate. Then only this substrate will undergo chemical reaction. Okay, this is this stage is for only for some time. This is a transient period, transient state or a transient period for a short while. Okay, and then it is produced into a product. So here is the definition is the difference in the average energy content of S from that of its transition state. This is the transition state. Here is here it is one part of the energy, say whatever the energy is there, that is its base level, and to take part in the chemical reaction to take place. Chemical reaction to takes place, energy level should increase. This is the transition state, energy at this transition state. This difference from here to here, here is some amount of energy, here this is the required energy from here to here. This energy is called activation energy. Whether it is correct, help to change a reactant into unstable transition state before they can be converted into a product. This is the transition state. In between, substrate change into product, but in between this from journey from here to here, okay, this is the transition period, and it is called the transition state. And this state is unstable. Say the substrate is stable, product is stable, but in between this is unstable. All right, this is unstable. So this help to change a reactant into unstable transition state before they are changed into product. So activation energy help it to change a reactant into unstable state. So make it unstable. The substrate is become unstable, and unstable it become by addition of some energy. Okay, that energy is activation energy, or the activation energy is the minimum energy required from outside to overcome the energy barrier. Say, this is substrate and this is the product. Okay, this is substrate, this is the product. And to come from the substrate to product, this much of energy is required. This much of energy is required. So, to change from substrate to product, there is a barrier of energy. There is a barrier, hindrance. Okay? This hindrance, and this hindrance is of energy requirement. So we call the energy barrier. So that energy barrier which is present in between substrate to product, this activation energy is breaking this energy barrier. Say, overcome the energy barrier. The minimum energy required from outside to overcome the energy barrier of reactant or to start a reaction. Here, I think you can see, say from our, yes, this diagram. Say this is the substrate. This side is, this line is showing the potential energy. So substrate has a potential energy here. This is sometimes called half Vmax, velocity of maximum velocity of the reaction. So if there is no enzyme, if no enzyme, then the energy from here to here, whatever the amount of energy, that much energy need to give from outside. this much. Okay, very high amount of energy is required. But if the enzyme is present, in that case, the requirement of energy is less. Okay, requirement of energy is less. So that so it is reducing the activation energy. Enzyme reduce activation energy. So it is this big, very big height, and say we are moving by a bicycle, no, or a car or vehicle, or you are walking, but by walking up to going up to this height, okay, you need a lot of energy. Then only you will reach to the other side. But if the elevation is reduced, say a road has been formed by a less elevation, okay, then you require less energy, and then you can say that okay, I can cross this border, is it? If otherwise, if this was the height, this is very difficult to cross, is it? So enzyme overcome the energy barrier. So activation energy, all these three are the definition, proper definition of the activation energy. Okay, all these are true for the activation energy. Difference between energy of the two state. This this is for unstable condition. This is required from outside to overcome the energy barrier. So we usually say that enzyme work by reducing the activation energy. Without enzyme, the activation energy is very high. So our answer is all of these are correct statement regarding the activation energy. Come to next one. Next one is some fill in the blanks. Fill in the blanks in the below statement by choosing the correct option. In a multi-step chemical reaction, when each of the state is catalyzed by the same enzyme complex or different enzymes, is called a dash. What is that pathway is called? In our skeletal muscle, okay, in our muscle, that mammalian muscles, under anaerobic condition, dash is formed. In yeast, during fermentation, the glycolytic pathway lead to the production of, what is that? For the A, B, C, here the words are given, but you have to write you have to choose the correct sequence and correct word. Metabolic pathway, acetic acid, ethanol. Glycolytic pathway, lactic acid, methanol. Glycolytic pathway, ethanol, methanol. Metabolic pathway, lactic acid, ethanol. Which one will be correct? Hello. Yes. Hello. Number 4. Same. You remember that any of the reaction, no, in biological system, usually there are many processes which is a chain of reaction. Say like glucose change into pyruvic acid. There are 10 steps, not in one step. This glucose change into pyruvic acid in 10 steps. So there are 10 different chemical reaction and 10 different enzymes are involved. Okay. So these are sometimes we call the enzyme complex, or it is say individual enzymes at every step. This type of chain of reaction we call metabolic pathway. Okay. What we call the chain of reaction is metabolic. So multi-step chemical reaction, and each one is catalyzed by a distinct enzyme. So this is called metabolic pathway. Here it is, it will be metabolic pathway. In the skeletal muscle under anaerobic condition, respiration, anaerobic respiration. In anaerobic respiration, what is the end product? Normally, end product is ethyl alcohol, is it? Ethyl alcohol, carbon dioxide. In anaerobic. In aerobic, it is, when it is aerobic, this one is carbon dioxide plus water. But anaerobic reaction also, this ethyl alcohol is produced when the fermentation or anaerobic reaction is carried out by yeast, Saccharomyces. They produce ethyl alcohol. In yeast, during fermentation, glycolytic pathway leads to the production of ethyl alcohol or ethanol. This is glycolysis, no, glucose to pyruvic acid, and then this one lead to ethanol if it is anaerobic. And another thing, in our skeletal muscle, this after the pyruvic acid is formed, then form the it will form lactic acid. Okay, it will form lactic acid. Here, in our skeletal muscle under anaerobic condition, lactic acid is formed. So our answer will be A, metabolic pathway, B, lactic acid, C, ethanol. Next one, inhibition of succinate dehydrogenase by malonate is an example of. Remember one thing, within our body, within the cell, many different types of chemical reaction is many different metabolic pathway is going on. So different reaction, different substrates are there, and each substrate or each enzyme is specific to a particular substrate. Okay, sometime what happen, some other chemical compound having the similar molecular structure, all right, similar molecular structure, they may compete for the enzyme. One another chemical, this is the substrate, but another chemical is there having the similar structure. So instead of this one will come and sit in the active site of the enzyme. Then what will happen? The substrate will not change. So this is a competition between the actual substrate and some other chemical compound which has similar molecular structure. So this inhibit the enzymatic reaction. Okay, the catalysis is inhibited. So this type of inhibition is known as competitive inhibition. This is known as competitive competitive inhibition. Here, inhibition of succinate dehydrogenase by malonate. Malonate is a compound which combine with succinate dehydrogenase and does not allow the uh, actual substrate to join this enzyme, succinate dehydrogenase. So this one is competitive irreversible competitive reversible inhibition. So competitive reversible in which way? That if plenty of the substrate you give there, then this will be reversed back. So this one is competitive reversible inhibition. All right. Then come to the next. Each enzyme shows its highest activity at particular temperature and pH. Enzymes are enzymes are very sensitive to very sensitive to temperature and pH of the medium. And it has their choice is not very wide range. The enzyme function within a narrow range, very narrow range of temperature and narrow range of pH, because pH is particularly why the enzymes are made of protein, and with the change in the pH, the structure of the amino acid, the protein changes. That's why their activity, catalytic activity is changed. So with temperature beyond 40 degree Celsius, the enzymes they denature. Denature means their 3D structure, that tertiary structure of the protein will break. Okay, unfolding will take place, and so functional activity will change. So due to beyond, when if you go beyond 40 degrees Celsius, they it denature. So this is, each enzyme shows a highest activity at a particular temperature and pH, and called the, this is optimum temperature and optimum pH. This optimum is the suitable one. The optimum temperature means at which the activity is maximum, at which the enzyme can work better or in its maximum capacity. So that is called the optimum temperature and optimum pH. Next question, select the correct statement from here. The low temperature preserve enzyme in a temporarily inactive state. Say this is also related to the temperature, effect of temperature on the enzyme. What happened within the living system? Within living system, to carry out the chemical reaction, it is enzyme should be, first thing that the enzyme should be active, and enzyme to be active, this is around 30 degree plus, but below 40. Okay, that enzyme work in a very nice way. If the temperature is low, the enzyme cannot work. Enzyme will remain in its good condition, but it cannot work. So it is preserved at low temperature, preserve the enzyme temporarily in inactive state. This is why that we keep our edible substances in the fridge, in refrigerator. Why do we keep? When our food or edible items, they are left outside for long period, they are spoiled. Cooked substances or uncooked one also. Okay, they get spoiled. But when it is kept under freeze or inside ice, then what happen? That the enzyme within the body of the those plant part or animal part, okay, they become inactive, so the chemical reaction does not take place, and that's why those foodstuffs or other things, they do not get spoiled. Okay, you have seen very common if you go to the fish market, then you what do you find? That they keep their fish, okay, in ice, along with the ice. That is keeping the temperature low, and they are storing, they can keep for several days without spoiling. But when we bring at home, we if we don't keep in the fridge, the fish will be spoiled. So this is because a low temperature preserve the enzymes, and high temperature denature or spoil the enzyme. Higher temperature destroy enzymatic activity because proteins are denatured by heat. This is also a correct thing. That is why at high temperature during the summer, I think you have heard many people die due to hot wave. So what happened at hot wave, the body temperature, outside temperature, that goes very high, and our enzyme system collapse. Okay, so we cannot remain living. Number three, activity of enzyme declines both below and above the optimum value. Yes. Say if 37 degrees Celsius, say this is the optimum temperature. All right. Then if the temperature we will bring down, say it is down, the activity of enzyme will go down. If we bring up, then also activity of the enzyme will go down. This is the optimum value or the optimum temperature. Sometimes this is 35, 32. Different enzymes has a different requirement maybe, but 30 to 35 degrees Celsius at this stage, the value is optimum where the enzymes work very nicely. Above and below, the activity decreases, and so all of them are correct. Select the correct statement, so all of them are correct. Here is another one. Which of the following statement is correct? Catalytic activity is lost when the cofactor is removed from the enzymes. Okay, come to another portion of the enzyme. Enzymes are protein, is it? Enzymes are protein. But many of the enzymes Some enzymes are pure protein, but many of the enzymes, they have a protein part, and along with this protein part, there is a non-protein part. The protein component and non-protein component. The protein component is called apoenzyme, and the non-protein component is the cofactor. So, an enzyme may consist of apoenzyme and cofactors. These together, this one together is holoenzyme. These cofactors are those enzymes where cofactors are there. This cofactor is very, very crucial, because if you take out the cofactor, then the enzyme, apoenzyme portion will not work. Cofactors are of three types. One is called prosthetic group, then coenzyme, and metallic activator, metal activator, or metal ions. This prosthetic group and the coenzymes, they are actually organic compound, usually organic, both. But difference between this one and this one is that this is tightly bound and permanent. This one is tightly bound, and the coenzyme is, it is transient. So it is loosely bound. Okay. Transient means temporary, for short living, and binds only when the catalytic activity is going on, means the reaction is taking place. After that, this portion will come out from the enzymes. And another thing is that these coenzymes, one coenzyme may be the part of several enzymes, because they are temporary. They go attached to the main protein part of the enzyme, and then after the reaction they come out. Okay, so one coenzyme may be the part of several catalytic activity of the enzymes. Those NAD, NADP, they are the example of coenzymes. FAD, right? And prosthetic groups are organic compound, other organic compound fixed to them. This metal ion, they are the metal ion. Many of the enzymes, they require the presence of some of the metal ion, like say sometimes zinc, sometimes chloride, in some cases magnesium, in some cases Mn. Okay, this become the part of the enzymes. So these are the cofactors. Now our question is catalytic activity is lost when the cofactor is removed from the enzyme. So as I told, cofactor is very very crucial, and with their removal, this, so this one is true, is it? Next is coenzyme nicotinamide adenine dinucleotide NAD that I told, and NADP contain the vitamin niacin. This is also true. Say this coenzyme again, coenzymes are organic compounds, but they again may contain some other compound also, and some of them they contain vitamin also. So this one is a niacin is a vitamin. Okay, this one is a B complex member. So this is also true. Biomacromolecules have hierarchy of structure. Biomacromolecules are which one? Biomacromolecules are like the protein, fat, sorry, the nucleic acid, and carbohydrate. Among the carbohydrate, the polysaccharides. They are the macromolecules. And you know, this particularly this polysaccharide proteins, they have hierarchy in their structure. Hierarchy means they have a primary structure, secondary structure, tertiary structure, quaternary structure, or some have primary, secondary, tertiary only. Okay, so different level of structural organization is there. This one is also true. This statement, proteins, by protein, you know that there is protein, there is a primary structure, secondary structure, tertiary structure. In DNA also there is primary structure, then there is the next is secondary structure, that double helical structure which is formed, that is the secondary structure of the nucleic acid. After that, further recoiling also takes place, that become the tertiary, and next is quaternary structure. Okay, then enzymes lower activation energy of reaction and enhance greatly the rate of reaction. This is enzyme function by lowering the activation energy. This one is also true. Say enzyme, this increase the rate of reaction greatly, sometime 10 millions time increase is there. Okay, or million time increase is also there. So this is true. Nucleic acid carry hereditary information. This is true. And passed on from parental generation to progeny. This is also true. So all of these are true. So our answer is this one. Okay. Next one question we will do. This is, let us see some one question from the matching. This one, here is this is based on the type of enzyme. Say the last question, type of enzymes. Type of enzymes is here in the column the name of the enzyme given, and in the second column the what type of reaction it carries out. And this classification of enzyme is based on the group or the type of enzymatic reaction. So there are one, two, three, four, five of these have been given, but there are six different types of enzymes are there. Okay, one has not been given here is lyase. So here the dehydrogenase, ligase, isomerase, hydrolase, transferase. From the name dehydrogenase, hydrogen taking out, it is what is called it oxidation, is it? So dehydrogenase is for the oxido reduction between two substrate, oxidation reduction. Ligase, ligating means sealing, that is two compound is joined by formation of the bond. So ligases linking together of two bonds, linking. Isomerase, isomers, molecular formula will remain the same, but their structural, positional, other things are little different. So, like glucose and fructose, they are isomer. Dihydroxyacetone and phosphoglyceraldehyde, they are isomers. So, isomerase is for the interconversion of optical, geometric, and positional isomer. Isomers are three types. This may be optical isomerism is there, geometrical isomerism is there, and positional isomerism is there. So this is isomerization. Hydrolases break the bond. Okay, it break the bond, or which called hydrolysis. When bond is break, water molecule is added. When bond is formed, water molecule is removed. So hydrolysis. By hydrolysis, they form the bond. Hydrolysis of bond, the breaking of bond by addition of water. And the transferase, transfer, it transfer groups. So this is group transfer. Okay, transferases, group transfer. So A with 3, which one? A with 3, say this is A with 5, A with 5, A with 4. So A with 3, only this one is there. It means this is the answer. A with 3, B with 4, C with 2, C with, yeah, C with 1, D with 5, and E with 2. So this one is the answer. Okay. So today